So to try to make this a little bit more fun and not too technically, I'd like to just tell you a few stories tonight. And the first thing I would like to do is to see if I can get you to come along and do a little thought experiment with me. So imagine you're in one of these things, one of these airport hangars, and imagine that while you're in that airport hangar, someone lays out all the individual parts of an airplane across the floor of the hangar. And imagine it was your job to assemble that aircraft piece by individual piece until you had a working airplane that safely transported people all across the globe. And imagine, are there any airline engineers in here? Any aerospace engineers? No? Okay, sweet, this works. So imagine I just gave you one of these old school flip phones, and I told you the only instructions you could receive was via this flip phone. And on the other end of the flip phone was an aerospace engineer that is the world's expert on that plane. So they know exactly how to assemble it piece by piece, but you could only send texts and you could only make phone calls to this engineer. You might imagine that's a little daunting and it might not be the most ideal situation, right? So you can imagine you're making a phone call and engineer Kim is telling you, hey, go to the mat, pick up part number 9691, grab this other one, try to put them together, but it doesn't fit too well, so you got to jiggle it a little bit this one way. And imagine just trying to have that communication just by voice. But then imagine, instead of the flip phone, we gave you a smartphone. And with the smartphone, you can now have a real-time video feed with that engineer. So if you're maybe having trouble putting two parts together, that engineer could physically show you how to put those two parts together. And if maybe you still couldn't put it together, you could maybe put your phone over the piece and the engineer would be like, well, if you turn it that way, and then. So the amount of information that this smartphone can convey is much more robust and it's much more nuanced and allows you to navigate this very complicated process perhaps a little bit more easily. And in addition to the video, maybe the engineer could also send you schematics. You could also access other resources online, right? So it's a much more robust, much more complicated type of communication system. Well, the reason why I'm mentioning that is because cells, all the cells in your body are constantly in active communication with each other. And a lot of times in disease, part of what's going wrong is that these cells are not communicating with each other in an effective way. There's something going wrong in this communication system, and if you could just fix that communication system, or one aspect of it, then you would be able to get rid of a lot of the symptoms of the disease. So there are two broad types of cell communication system. And the analogy I like to make is the one that we all know and love in biomedical research. It's sort of like a flip phone. It's very robust. It's a little bit more on the simple side, but it works very, very well. But in the last few years, there's been a new discovery, and it's a new type of cell-to-cell communication system that's much more robust. So when our lab and other labs try to develop stem cell therapeutics, instead of trying to focus on this old school communication system to develop new therapeutics, we can use this new type of cell communication system. And hopefully that idea will be a little bit more crystallized as I start to explain what the communication systems actually are. So, and if anybody has any questions, feel free to stop in the middle of this and shout it out. I'd like this to be as productive as possible. And I think we have a microphone for Q&A a little bit later. But this old school communication system is fairly straightforward. These little individual proteins can get secreted outside of a cell and then float around in the space outside of the cell, in between cells, and will float around until it comes into contact with the receptor that recognizes specifically that one protein. And of all the proteins in the human genome, we know that only 1% of the genes in the genome, human genome, encode for this communication system. Just 1% out of all the thousands and thousands of genes, just 1%. And we know that it's just 1% because these proteins have to have a couple of special things about them. They have to have a special tag on them that tells the cell to, hey, secrete me outside of the cell. And during that process, when the cell is making that protein and shuttling it along its way to secrete it outside, it also throws a bunch of sugars, a bunch of carbohydrates on the outside, so it can sort of survive on the outside environment a little bit better. But this new communication system gets around this problem, right? So how can you use the other 99% of the human genome to communicate to another cell with if all these other proteins don't have those two special things? They do not have the tag that tell it to get secreted, and it doesn't have all these fancy sugars added to the top of it so it can survive the outside environment. How the cell gets around this, which is something we're just now recently discovering, is it'll secrete these little bubbles. And these little bubbles house that other 99% of the human genome, the other thousands and thousands of proteins encoded in our genome. And now it's these bubbles that will float around until they come into contact with another cell and then deliver its contents. So all those proteins inside that bubble, which I'm going to call exosomes, never really see an environment that looks like it's the outside of the cell because it's encased in that bubble that was made inside of the cell. And what is inside of these little bubbles are a bunch of things that we normally don't think of as being transferred from one cell to another cell. And I'm not going to go into a lot of the technical detail, but I think it's suffice to say that it's thousands upon thousands of cellular components. And when we say the old school communication system just uses 1% of the human genome, and this new system uses 99%, even that's an oversimplification because there's lots of things inside of the cell that are highly functional that are not encoded in the genome. So for instance, non-coding RNAs, which help turn genes on and off, and other things like metabolites and lipids. So this new communication system is around at least 2 orders of magnitude more complex. And so why am I so interested in how cells talk to one another? Well, here is an electron micrograph showing some of these bubbles coming off of one particular cell type called a mesenchymal stem cell. And mesenchymal stem cells are a very special kind of stem cell that come from your bone marrow. You can also get them from other tissues, but the canonical, the most well-characterized come from your bone marrow. And these cells have a bunch of interesting properties, the most important of which is that they like to heal damaged tissue. And because of this, there's currently several hundred clinical trials going on trying to test these mesenchymal stem cells out for a bunch of different diseases. And each one of those clinical trials, by the way, is millions and millions of dollars, and there's hundreds of the clinical trials. So you can imagine the amount of resources that are currently going into this technology to try to develop them as a new therapeutic. And just to give you a little further insight, some people actually think this is a paradigm shift in how we are going to do medicine in the future. So it started with small molecules, so this is like Tylenol, chemotherapeutics, small chemicals, although I don't like using that term, because everything's a chemical, that get injected into a patient or somehow taken up. Then we had this big revolution in therapeutics called the biotechnology revolution, happened in the late 70s, early 80s, really mushroomed in the 90s. And this is basically if a patient is missing a protein, that it needs to be happy and healthy. So for instance, yeah, we don't have to get into examples. It's basically teaching bacteria to be little manufacturing plants to make that human version of that protein that we then purify from the bacteria and inject into the patient. But now the thought is, hey, well, instead of injecting into a patient a small molecule drug or a purified human protein that the patient's missing, the idea is, hey, maybe we can inject human cells into the patient, and now those cells are therapeutic. And so that's this newer technology that's coming along that is what's comprising these clinical trials. And so why I'm interested in the cell communication system is very pertinent to how these stem cells work. But I think it's a little confusing for a lot of people when we try to talk about our therapeutics with these adult stem cells because it's very counterintuitive, especially if you already know a little bit about stem cells. It's not very intuitive at all. So before I tell you how they work, I want to tell you how they don't work. Like, hey, how they don't do it. So what the heck are stem cells? So just like a brief overview, I'm sorry if this is too much review for some of you, but if we take a very immature stem cell, like an embryonic stem cell, which is mostly what you hear about in the mass media, and so this is what people are most familiar with. This cell has a bunch of interesting properties, two of which are the most important. One of the things it can do is it can go through something called asymmetric cell division. So you have a whole cell, and then it divides into two cells. One of those cells stays as an embryonic stem cell, and then the daughter cell will now mature or differentiate into a new cell type. And what happens as you go from an embryo through adulthood, there's a series of steps where that cell keeps going through asymmetric cell division and keeps getting more and more specialized until you get to all these cells, which are what comprise the vast majority of the mass of your body, which are these quote-unquote somatic cells. And there's two interesting properties about these cells. One of which I just mentioned is that they're highly specialized. So for instance, one way that I'll describe it sometimes is you can imagine this is a little child that maybe hasn't gone to school yet, and so it doesn't have any specialized education. But maybe we live in a different country with an unusual education system, and when they're really young, we either send them to a math and science school, an arts and humanities school, or a physical activity, they're going to go to the Olympics or something school. And then the next step, they get a little bit more specialized and a little bit more specialized until finally we have a chemical engineer that creates crystals for solar panels, something uber niche. So the vast majority of the cells in your body have highly specialized functions that all the younger versions of that cell can't perform. The other interesting thing about these cells is they don't divide. So if you lose a neuron, your other neurons are not going to go through cell division and give you a new neuron. Some of you now might be thinking, hey, Jonathan, I don't think that's 100% accurate. Are you sure? Because say if I have a cat and the cat scratches me and takes off some of my skin, my body will heal that skin. Where does that skin come from if these cells don't divide anymore? Well, where they come from is actually from these cells. So these cells are a little bit more immature. We'll call them progenitor cells. And a progenitor cell is capable of making, for this particular example, say a bunch of skin cells. But it's only able to divide so many times, and then when this gets tired, there's a cell before that. In real life, this flow chart is much more complicated and in much more detail, and imagine many more layers. So a couple layers before the progenitor layer are these adult stem cells. So right now, as an adult, you have a bunch of different kinds of stem cells all throughout your body. There's not a lot of them, and each of them have a specialized function, and most of the time they just sort of hang out and don't do a lot, and only when they're needed do they make another one of the progenitor cells. And then it's the progenitor cells that will make these specialized cells. So when scientists first discovered these mesenchymal stem cells from the bone marrow, they could see that there were a stem cell. And since they got them from the bone marrow, they're like, hey, I wonder if they can maybe differentiate into a bone cell, an osteoblast. So they sprinkled a cocktail in the dish, and they're able to see, yeah, these cells are able to become bone. They could also show that they could form cartilage, and they could also form fat cells. And so then they started to think, like, hey, well, we have an adult stem cell. We don't have to worry about messing with embryonic stem cells or taking them through this education process. They're already halfway through the education process. Maybe we can just inject these cells into different animal models of disease, and maybe those by doing that, they'll take the place of the dead and dying cells. So the thought process on that, on the sort of canonical stem cell therapeutics, is that you have a cell, it's a little bit immature, you're going to inject it, say, into someone with a heart attack. So in a heart attack, the blood, one of the arteries feeding a part of the heart gets clogged. And your heart's working hard. It's a muscle that's constantly contracting, so it needs a lot of oxygen, needs a lot of sugar, and a lot of factors to keep it going. So if you block off the blood supply, you're blocking off the oxygen and all the nutrients it needs, right? So you can imagine bad stuff happens. And one of the things that happen is you get a little infarct. And the infarct is a bunch of cells that are in a little circle that gets fed by that one particular blood vessel end up dying. And so what the scientists thought, since they didn't know exactly what these mesenchymal stem cells could differentiate into, they started injecting them to a bunch of different animal models with the hopes that the cells would take cues from their environment. So cells are always in constant communication with their environment, and they're always sensing their environment. Very much how I'm sensing in my environment. I have bright lights on me. I'm up on a stage. I have an audience in front of me. That gives me some sort of context of what I should be doing, as opposed to, say, if I'm at a basketball game with my guy friends and we're having some beers. I might be acting slightly different in that context. Cells do the exact same thing. So for instance, when they're trying to see if they could differentiate into different cell types, that's exactly what they did. They gave it little communication signals that said, hey, maybe you should think about turning bone, and it would turn into bone. So when they injected these cells into these different types of diseases, cardiovascular disease, kidney disease, and liver disease, a bunch of different diseases, what do you guess would happen? I'll tell you what happened, nothing. Or at least that's the way it seemed when they first injected them. So those cells, even though they should have been in an environment where they're taking cues, so for instance, let's look at the heart attack model, they could see they're in cardiac tissue. They could see their cells dying in front of them, and these cells naturally like to help out. You would think, the thought was originally, that they would turn themselves into a cardiac cell, a cardiac muscle cell, and take the place of the dead and dying cells. And they marked these cells before they injected them so they could keep track of where they went and what they turned into. And to this day, whenever they inject these mesenchymal stem cells into a bunch of these different animal models, they do not differentiate to any appreciable extent to any different cell type. They just stay as a mesenchymal stem cell, and they don't even stick around that long. They just kill themselves after a few weeks. You can argue about how long they stick around, but it's sort of like geometric decay. As soon as you inject them, they just sort of fall off a cliff in terms of how many are around over time. So how do they work? How do they have any benefit? So at first they were very disappointed because they didn't differentiate into any particular cell type, but when they went back and looked at the data, so for instance, if they gave a rat a heart attack and they treated that rat's heart with some of these mesenchymal stem cells, even though they didn't change into a cardiomyocyte, a heart muscle cell, they still seemed to have a positive effect. And what they attributed that to was, let me see if I can play this little video while I'm talking. Or not, it's okay. What they attributed to was these cell communication systems. So what these cells, oh, it's playing. Okay, sweet. I don't know how that happened, but I'll go with it. What they do is when they inject these mesenchymal stem cells, they literally just secrete out these little communication signals to the surrounding tissue and say, you know what, I know you're trying to fix this little infarct zone. Let me help you and direct the traffic a little bit and try to help you fix it a little bit more efficiently. And so, for instance, my boss, Jan Nolta, who's the director of our Stem Cell Institute at UC Davis Medical Center, likes to call these mesenchymal stem cells the paramedics of the body. So they just naturally like to repair damaged tissue. So if you have an open wound, there'll be signals sent out from that wound, hey, we're stressing, and some of your mesenchymal stem cells will migrate towards that wound and help repair that. So in one way, it makes a lot of sense that we're taking bone marrow from somebody, isolating these cells that like to help, and then we're injecting them into the site of injury, right? Because that's what they like to do. But so for the majority of the time that these cells have been around, it's been known that they facilitate this tissue healing process through cell-to-cell communication. But what they thought the cells were using was just the old school flip phone, right? The cells were sending, these mesenchymal stem cells were sending texts, and they were making phone calls to the surrounding tissue, and that's how they were facilitating the tissue healing. But it turns out there's this new type of cell communication system that we know that these mesenchymal stem cells use, and it's mediated by these little bubbles, which here sort of look like deflated soccer balls, but in real life, they're spherical. They just look deflated here because this electron microscopy methodology makes you dry out the sample, so they're deflated because they're dehydrated. So you might say, hey, well, that's fine and dandy that these cells secrete these little bubbles, but are they actually doing anything? Are they functional? You know, maybe they're just trash that the cells are trying to get rid of. Oh, and incidentally, this communication system we now know is evolutionarily conserved all the way down to bacteria. And what that means is Mother Nature has thought that this is an extremely important thing, that we're going to keep it from back when we were not quite as evolved through now. So Mother Nature has thought this communication system was super important to keep it around, basically. And what they have shown is, you know what? Instead of injecting these mesenchymal stem cells into these different animal models and seeing a benefit, what if we just scooped up all of the bubbles that the mesenchymal stem cells secrete into the media that we grow them in and just isolate out just the bubbles, just the exosomes? And what if we just injected the exosomes into all these animal models that we see the mesenchymal stem cells are helpful in? And what they see is, sure enough, yeah, it seems to have the same benefit as if you injected the cells themselves. So, excuse me. I'm not going to go into the details, but just to give you a few examples. In a model of acute kidney injury, the exosomes from mesenchymal stem cells, these MSCs, are able to induce tissue healing. And also in a model of liver damage, it's able to stop fibrosis, which is scar tissue, which is really bad. It's able to prevent that. And in a couple of different cardiovascular models, it's also able to have a good effect, these exosomes. So for instance, here's a model of, someone in the audience was asking me about peripheral arterial disease before I started. This is a model of peripheral arterial disease. And these colors have significance. The warmer colors indicate blood flow, and the cooler colors indicate lack of blood flow. So here you can see they severely limited the blood flow in the leg of this animal. And here they injected, over here they injected the MSCs, the cells, and here they just injected the exosomes. And you can see the exosomes were able to help restore the blood flow in a comparable fashion to the MSCs. So one way to think about this is, instead of injecting the cells, and the cells being little factories of secreting all these healing factors, why don't we just scoop up all the healing factors into a concentrated injection and just inject what's doing all the healing, right? And here is a heart attack model where here's a cross-section of a normal healthy heart. And here is an extremely severe heart attack model where the infarct zone is this thin blue material. You can see a large part of this rat's heart has atrophied and turned to scar tissue. And if you inject MSCs, you can see that there's much less scar tissue. And if you inject the exosomes, or these bubbles from the MSCs, it's able to have a beneficial effect as well. And so this is not a completely academic discussion either, right? So a lot of times things that work well in animal models, for a lot of different reasons, don't translate well into patients. But the first patient has already been successfully treated with these exosomes from mesenchymal stem cells. And it's a patient in Germany. And this patient had something called graft versus host disease, which is a transplant rejection. And this particular patient had a bone marrow transplant. And even though they try to match the tissue very well, you normally get some sort of immune reaction in the patient. Sometimes that immune reaction is manageable with drugs, and other times, in a more rare instance, it's not, and the transplant actually ends up killing the patient. So this patient was refractory, which means they weren't responding to any of the standard treatments for graft versus host disease, and this is what they look like. This is all the same patient. So you can see they're not doing so great. And so it was thought that this patient was going to be terminal because they weren't responding to any of the drugs, and the patient was just sort of spiraling downhill. So the Germans' version of the FDA allowed a compassionate use of an experimental drug. And the experimental drug they decided to give were exosomes from mesenchymal stem cells, which makes a lot of sense because these mesenchymal stem cells have been shown to be beneficial in diminishing this hyperimmune response. And so this is before treatment. This is after the end of the fourth and final dose. And this is 16 weeks after the final dose. And you can see it helped this patient. So it should be taken with a grain of salt. It was just a one-off. It wasn't a clinical trial. It was just a compassionate use, but at least shows that it's feasible that these exosomes are able to be beneficial. So I am interested, when I joined Jan's lab about 5 years ago, is right around when I first started to hear about these little bubbles, these exosomes. And it's right around the time that we all started to realize, like, oh, wow, they might be actually playing a part in a lot of the things we study. So I'm interested in neurological diseases, and I'm also interested in cardiovascular diseases. And the initial studies I did involved cardiovascular disease. because it was a little easier to work with the models, basically. But I'll also mention neurological diseases in a second. So sorry, I'm getting ahead of myself. So we can see what these little spider web-like things are blood vessel formation in a dish. And if we don't give the cells anything, the cells that make up your blood vessels, because we have to plate them on this special gel. That special gel we put them on, it's sort of a signal for them to induce themselves to aggregate into these long, thin tubes that become your blood vessels. So even just giving them the gel that they sit on induces some amount of angiogenesis. That's this guy right here. But if we give an increasing dose of exosomes, we can see that it induces these cells to grow more and more blood vessels. And this is good for both heart attack and for peripheral arterial disease, because basically you have a plumbing problem. You have a blockage in the pipe, and this helps reroute around the blockage to restore the plumbing. And I already mentioned this, that yes, the exosomes can reintroduce blood flow. So now that our lab and other labs have shown that these exosomes, these bubbles, are able to induce tissue healing, one of the big questions is now, well, how are they doing it? So it was right around this time I got to work with this really great lab and this PI out at the Karolinska Institute, which is in Stockholm. And so for those of you who don't know, this is a very prestigious institute. Some people call it the Harvard of Europe. It's been around for a couple hundred years, and it's where they give out the Nobel Prize every year. And this gentleman, Janne Lettio, is the director of their something called their clinical proteomics program. And basically what he's done is this idea of proteomics, which is basically trying to quantify all the proteins, all the genes in your cell and how turned on or turned off they are. That technology has been around for a few years, but normally with the methods that people normally use, you can only look at a few of the proteins at a time. But Janne was able to develop this new fancy version of proteomics. And now with his technique, you can look at comprehensively all the different levels of all the proteins and how they change in the cell over time with this very fancy technique. So I was lucky enough, this is me smiling and be very happy because I'm living in Stockholm right now for a few months during this picture, which was a good time, and I learned a lot. And what we learned is that, you know what, yeah, if they have a bunch of tissue healing properties, it makes sense that they are chock full of a bunch of proteins that help facilitate, that you would think would help facilitate that process. And that's exactly what we found. And we recently published this in a paper in the journal Stem Cells, which is a very nice journal, and we're very proud of it. Here's the title of it. I can send it to Dr. Sproles if anybody's interested. And it's got a lot of buzz going around it. So there's this score called the altmetric score, and it's basically a score of how much people are talking about it on the internet. And this is a pretty good score. So we're in the top like 97th percentile or something like that. So we're excited we got this out. And basically what this shows is not only can we show what's inside of the exosomes on a protein level and what might be mediating this tissue healing effect, but we can also show that part of it goes through this one group of proteins that's involved in something called NF-kappa B, or nuclear factor kappa signaling. Okay, so ignore the fact that that looks complicated, because I'm just going to gloss over this super quick, I swear. What we have here is a bunch of, you remember how I said I was interested in neuro stuff? Well, what this paper talks about is all the good stuff for cardiovascular disease that are in the exosomes. But when I was analyzing this data, I also noticed a bunch of stuff in the cells that seemed to be good for neuro stuff. So here we have something called a signaling pathway map. And basically this map shows a lot of the proteins that we think are involved in this thing called efferent signaling. And if you see a triangle or circle or square that's either pink or green, that means we saw it inside of our exosomes. And if it's just completely empty, it's all white, that means we did not see it in our exosomes. So what you can see is inside the exosomes, we see a whole heck of a lot of proteins involved in this efferent signaling inside of the exosomes. And so what that means is when we deliver these exosomes, these bubbles, to neurons, they're going to likely induce this efferent signaling. We see a similar thing with something called axon guidance, something called paxillin signaling, integrin signaling, actin signaling, phagosome maturation, and Krebs signaling, and a bunch of others. the nutshell take home of all that data is there's a whole heck of a lot of proteins that are inside of the exosomes that would make you think that, hey, maybe they would be beneficial in certain neurological diseases where the brain cells are dying a little faster than they normally should. So we developed a collaboration with a lab in Michigan to see if our exosomes might be beneficial in a neurological model. In a rat model of a neurological disease. And the disease we decided to look at was something called stroke, ischemic stroke, which is the common kind of stroke that everybody gets. And it's basically where there's a blockage in one of your blood vessels inside of your head, and you starve a good chunk of your neurons inside of your brain for oxygen and nutrients. And same thing with the heart attack. The cells don't like that because they're very metabolically active, so they start killing themselves. And you get the same thing, you get an infarct zone inside of your brain. And as the cells stress out more and more, that dead zone grows larger and larger. So you can imagine that's not good, right? Say it the least, right? And so, for instance, if you have an infarct, if the blockage occurred on the left side of your brain, and nice, I like the ringtone. On the left side of your brain, if you get this dead zone on the left side of your brain, the right side, it controls the right side of your body. So if you have a severe enough infarct, you'll start to have trouble controlling the right side of your body. You'll have trouble with the motor skills on that side, right? And as the infarct gets worse, you'll have like more and more trouble controlling it. And if you could give the rat or the patient something that would help out, then they'll regain that movement back to where they can use both sides of their body equally well. So we have to use measures like that, how the rats are physically moving, because we can't really ask a rat, how are you feeling? What's your thought process like, you know? So here what we see is right after, a couple days after we give the rats the stroke, if the bar goes down, which is they did not get anything to help them out, the infarct seems to be making the rats worse and worse. They're having more and more trouble controlling that side of their body over time as that infarct is probably growing bigger and bigger over time. But what we see if we give it exosomes from MSCs is that, yeah, they actually regain some of that physical function back on that side of their body. And this was actually a happy accident in terms of there was, interestingly, a miscommunication with this student that was running this model for us in Michigan. We wanted them to deliver our therapeutic 24 hours after they induced the infarct in the rat brain. Give me 1 second, I'll get to you. Somehow the student mistakenly heard 24 hours to be 72 hours. So instead of delivering our therapeutic, which would be optimal to deliver no later than 24 hours, they waited 3 whole days instead of just 1 day to deliver it. And even, which is good, it was giving me a little bit of an ulcer before we found out the results, in all honesty, because 3 days out, you're thinking a lot of the damage is already done and it's not really going to recover. But even 3 days out after the stroke, it's still able to induce a positive effect. Yeah, there's a question in the front. So, sorry if this is a little morbid for some of the people in the audience, but it's we use the classic model. It's called MCAO, and they basically open up a hole in one of the major arteries in the mouse, and they they feed a little thread in that artery. And the artery that they inserted in is, say, yay big, and the thread is maybe so big. Right? It's way more, it's tiny enough that blood flow can go, pass over the thread. But as you stuff that thread higher and higher up the brain to its middle cerebral artery, which is why it's called MCAO, the O is for occlusion, you eventually get a point in the artery where it's small enough that that thread is so big that it actually completely blocks it. And so they leave that thread in there for about a half hour, and then they take it back out. So it only really has the infarct, or has the occlusion, has the blockage for about a half hour, and then they take it back out. Sometimes you can leave it up there a little longer if you want more of a severe model, but that's basically the model. So this bodes well for us. We're really excited about this data. This is just our first pilot study, but it looks pretty good. And we have another model that we recently, this is like hot off the presses, like a couple days old. We have another collaborator at the Med Center, and she's very interested in retinal degeneration, which is, again, another very common problem. And what you can see in retinal degeneration, one of the things that indicates that the retina is degenerating is that, you remember how I showed you on one of the previous slides that little web-like network, and I said it was blood vessel growth? Well, these images are picturing the same thing. So if you can imagine your eye is a sphere, right? And if I make my hand as the back of your eye, there's a major nerve bundle that comes in the back. And that nerve bundle, it looks like this middle of the spider web, and here it's off screen, here it's off screen. And what comes out of that nerve bundle is also a bunch of vasculature. And what's being imaged right here is the vasculature. And over here is a healthy rat. And you can see that these big giant guys are the sort of the major arterioles that are feeding blood into that eye. What's a little harder to see are all these little wispy spider web-like things that are in between the major blood vessels. These are called capillaries. And it's really the capillaries that actually feed your tissue. The main arteries, not a lot leaks out of them, but when you get down to capillaries, the capillaries are leaky enough that a lot of the oxygen and nutrients can sort of leak out. So this is what it looks like in a healthy eye. If you induce this one particular kind of retinal degeneration, which involves lack of oxygen, you can see a lot of the little spider web-like things, a lot of these little capillaries end up going away. And that ends up leading to the retina degenerating and loss of sight. So if you give the rat, these exosomes at the same time, you can see it salvages. It keeps a lot of these spider-like capillaries. So if we go from here to what is in the disease state, this is what our exosomes are able to do with this particular disease. So we get asked a lot of times, I think the gentleman that was asking about peripheral arterial disease, I'm not sure if he's still in the audience, but our lab makes therapeutics, right? Stem cell therapeutics, and we try to take them through clinical trials. So we have 2 products right now that are just done with the preclinical stage, which is sort of a clinical trial in animals before you go into humans in phase 1. But this is another clinical trial that is going on at the hospital at UC Davis Med Center, and it's already in people that is in collaboration with our lab. And they've already completed phase 1. So they've already tested the safety out, not only in the animal models in the preclinical stage, but they've already tested it out in I think about like 20 patients or so, and they got good results. So they just put in a grant, because these clinical trials are expensive, to pay for phase 2 of the clinical trial. And the investigator, the PI at the Med Center, Susanna Park, was so excited about this data that she decided to try to add on to see if the FDA and the funding agencies will let us add on these exosomes as an adjuvant therapeutic to their cell therapeutic. So basically an adjuvant is something that enhances the effect of your primary therapeutic. So we just submitted that grant a couple of weeks ago, so fingers crossed it goes well. But for me, just trying to be an up-and-comer at the Med Center, it's pretty exciting to be involved in sort of heady stuff like this. So what the heck does a mushroom coral have to do with this communication system? Well, I think it's cool, and I think it's sexy, if I do say so myself, that these little bubbles from these mesenchymal stem cells are able to induce tissue healing. Let's see how I'm doing on time. Okay, I should wrap things up. But that's not really the sexiest thing from my point of view. It's great that they facilitate tissue healing, and it's also great that if we just inject these bubbles, these exosomes, instead of the cells, maybe that's beneficial because we're delivering a more concentrated form of the therapeutic, right? So if we inject the cells and they immediately just start dying, you imagine, and you think of each cell as a little manufacturing plant, you're losing all your manufacturing capabilities right after you inject all the cells. Not great. So why don't we just have the manufacture in them outside of the body, all these therapeutic molecules, and then just concentrate those and inject those. So that's what I've talked about so far, but that's not, for me, the real kicker. The real kicker is, if you remember, I said the old school communication system uses about 1% of the human genome, and this new communication system at least uses 99% of the human genome, and maybe other factors in there like non-coding RNAs, which help turn genes on and off. So if you think about that for a second, it opens up a lot of engineering doors to develop novel therapeutics. So for instance, you know how I said we had 2 products that just went from the preclinical phase that we're hoping goes into phase 1. What our lab and other labs have tried to do is say, you know what? Okay, these mesenchymal stem cells work really good at healing damaged tissue in some of these diseases, but maybe we can make them a little bit better by genetically engineering them. So if, we rewind the clock back a few years, and they have all the smart people in the room, and they're all scratching their chins, like, what should we tell the cells to make more of? They have to make a decision, right? What's going to be the best thing to tell the cells to make more of to have more of a therapeutic effect? Well, back then, all they knew about was the flip phone technology, right? So they're thinking of texts and they're thinking of phone calls. They're thinking of 1% of the human genome. Versus now, if you know that these cells are secreting these vesicles, now your engineering options, instead of your library from which with you have to choose a therapeutic to tell the cells to make more of, instead of just a few hundred options from 1% of the human genome, you now have the other 99% to ostensibly choose from. So it opens up a lot of engineering doors. And that's just on a gene level. There's also things we can deliver, package in these things, which I'm getting ready to show, which brings me back to the mushroom coral, that don't normally belong in the cell that could still be of potential benefit in treating patients. So for instance, the reason why this is up here is because not only is it really pretty, but scientists have discovered that this mushroom coral has a protein in it that likes to fluoresce red. And they've taken that protein out of the mushroom coral, and they've engineered it a little bit, and now they've turned it into something that they commonly call TD tomato. What TD tomato is just a fluorescent protein. So for instance, if you want to track cells or track something, and if you're able to package this protein inside of it, then it'll glow bright red, so it's easy to follow where the protein is, right, in real time. So if we genetically engineer our mesenchymal stem cells to overexpress this TD tomato, this fluorescent protein, sure enough, we get a lot of fluorescent protein inside the cells. That's nothing new. That's like technology that's been around for at least a couple decades, right? What is new is that we can show that we can package not only the protein inside of the vesicles, right? We can also package the mRNA. And if you give me 5 seconds, I'll tell you why that's super sexy from my point of view. Is if you deliver a protein to a cell, you have, let's say, 1 unit of function. Say, for this case, it's a red fluorescent protein. It'll give off so much fluorescence just for that one protein. But if you deliver 1 mRNA that encodes that protein, that 1 mRNA will make hundreds to thousands of copies of that protein. So you can see it's several orders of magnitude more potent to deliver 1 mRNA as opposed to delivering 1 protein, right? So what this shows is, hey, if we overexpress this TD tomato inside the cells, we genetically modify it. The cells will then make the mRNA, which then gets translated into the protein, to the fluorescent protein. We can not only see that we get a little bit of signal that the exosomes, if we just take out the bubbles and put it on a different cell type, we can see that the exosomes are delivering a little bit of the protein. So you can barely see a little bit of redness right there at 8 hours after we deliver the exosomes. But if you wait 72 hours, you can see the signal is much, much higher. And what this likely means is that we're delivering the mRNA as well, and that the cell is now able to make hundreds and thousands of copies of the protein, which is why it looks so much brighter. So now what we're trying to do, so this is just a proof of principle. No one's interested in using this protein as a therapeutic. It just allows you to track how your genetic engineering techniques are going. So this lets us know that we are able to genetically engineer the exosomes. And so what we're actively doing now is trying to enrich them for certain therapeutics that we think will be beneficial for both cardiovascular and neurological diseases. And not only are we able to deliver the protein and the mRNA, but we can actually deliver the plasmid, or the DNA. So if you deliver the DNA is going to make several hundred to several thousand copies of the mRNA, and each one of those mRNAs are going to make several hundred or several thousand copies of the protein. So that is, it bodes well for developing more potent therapeutics using this new communication system. So I'll just wrap up. There's a lot of Bridges students in the audience, or at least some. And we love the Bridges students here. So for those who don't know, there's a really awesome program here at UC Davis called the Bridges Program. It's funded by our state Regenerative Medicine Institute, CIRM, California Institute for Regenerative Medicine. And it's basically a master's program where they get it specialized in stem cell research. So I have 6 members on my team, 3 of which are Bridges students, which have worked out really awesome for me, and I'm super stoked, which is part of the reason why I wanted to come and talk to everybody and how I know about this program. And so I want to sell us a little bit and basically just say, hey, we have this very fancy institute at the Med Center. It's called the Institute for Regenerative Cures. There's lots of cool and fancy things inside of this Institute for Regenerative Cures, including something called a GMP facility, a good manufacturing practice facility. So if you ever want to inject a therapeutic into a patient, it's got to meet extremely high standards of purity and cleanness. And we are super, super lucky here at UC Davis to have one of these facilities that specializes in cellular therapeutics and also viral vectors for gene therapy. And so our team actually isolates our exosomes inside of this GMP facility, so they're GMP grade, so an extremely clean, extremely pure form of the therapeutic. So yeah, this is a little shout out to my team members who helped me generate all this data. The 3 Bridges students are Zelenia Contreras, Madeline Hoon, and Missy Pham. These 2 are from Humboldt State, and Missy's from San Francisco State. And here are some of our collaborators on the project. So, and here's my email address if anybody has any questions, but I think maybe they've allotted a couple minutes for questions here. And I think there's a microphone in the front, so if anyone's brave enough to come up and ask a question, feel free to hit me. That was a good question earlier. Thank you. I appreciate it. And also a big shout out to the IT guys who were like up here. We literally had to go through 4 different laptops and 2 different microphones to get everything set up. So thanks for sticking with it.
Is it on? Okay. What kind of I'm projecting, insurance companies and things like that when it comes to cellular research and also for these therapeutic models that you're thinking about. What do you see in the future?
That's a good question. So in New Zealand, Canada, and Japan, insurance is already reimbursing mesenchymal stem cells for a disease. Remember the patient I showed, the graft versus host disease? Mesenchymal stem cells have already been approved in those countries for graft versus host disease. And so it's working well enough that it's passed their clinical trial pipeline. So it's literally a drug you can buy and you can get a prescription for, and it's reimbursed by insurance. And since it's been working so well in the clinical trial so far, they're trying to fast track some of these mesenchymal stem cell therapeutics through clinical trials. You need to wait until the clinical trials are done and over with before you really know. Everyone always has a lot of excitement about new therapeutics. It's easy to get caught up in the hype. So you really have to wait to see what the data looks like. But as the data is coming out and it's looking good, that's not stopping the FDA or insurance companies or big pharma from acquiring those companies and taking it to market. So. Mm-hmm.
When you were using the mouse models, how was the exosomes delivered? Was it a local injection or was it through an IV delivery method?
Yeah, that's a good question. The ideal delivery method is still open for debate, but we did a localized injection. Okay. So for instance, for both the stroke model and the eye model, it was a localized injection. For the stroke, it's an intracranial injection, so they drill a tiny, tiny little hole, which sounds crazy, but they do it in hospitals all the time for certain therapeutics, and take a very thin needle, and they try to inject it around the area where they think the infarct is. And the same thing for the eye model. They take a very small needle and very carefully inject it in the back of the eye.
Okay. And was it just one injection, or was it multiple injections over a series of days?
That may be the better route to go eventually, and it'll be a little context dependent depending on the disease, but just as like a proof of principle, we just want to know if, will one shot even do anything at all? So what I showed was just one injection.
Okay.
Oh, this guy looks serious. I bet it's going to be a good question.
Can you tell us how the Institute is funded?
That is also an awesome question. Thank you for asking that. We are a super duper crazy, crazy lucky here in California to have our own funding agency specifically for stem cell research. It was a ballot measure about 15 years ago where California decided they wanted to put several $billions to turn California into a mecca for stem cell research, and that's exactly what it's done. When they made that program, they didn't know how good of an idea or not it was, so it's getting ready to sunset in about 6 or 7 years or so. So they're hoping by the time that takes place, we will have made enough advances that the residents of California will think it's a good idea to keep investing in it. So not only is it great because we have our own funding agency, it's particularly what they fund, not only in terms of stem cell therapeutics, but the level of the research. So there's the basic biology, and then you try it in animals, and then animal clinical trial. When you get to like the animal clinical trial in phase 1, phase 2, some people call that the valley of death. That's because our other national funding agencies, like the National Institutes of Health and Big Pharma, have just sort of like stepped away. It's extremely hard to get funding for that. They're very risk adverse. And what CIRM has done an excellent job of is funding this valley of death. So that's the vast majority of what funds us and a lot of the other people at our Institute for Regenerative Cures. So yay, CIRM. And it's also what paid for this Bridges program, by the way, which is this awesome master's program here at Sac State and a couple other universities.
Was there actually an agency created for that measure?
Yep. They have headquarters and everything. They just moved from San Francisco out to Berkeley because San Francisco rent prices were getting too expensive. But yeah, it's a big old juggernaut. And they recently just changed guard in terms of who is running it. But yeah, it's a bona fide agency. Yeah, and they even have like a helpful blog if you're interested in stem cell therapeutics. If you just look up CIRM, C-I-R-M, you'll get to their home website, which has a helpful blog of a lot of the stem cell therapeutic stuff that's going on nowadays in California.
Regarding stem cells for stroke patients, do they have to have the injections within the 3 days, or can they do it at a later date?
That's a good question. Yes and no. So the earlier that you're able to deliver a therapeutic, the better it is because then you limit the amount of damage. So after the original stroke, the tissue keeps stressing out more and more, and you get an increasing amount of the infarct of the cell death. Eventually that sort of plateaus and it sort of limits itself. But if you could limit that therapeutic, the better it'd be. So part of the reason why I was so frustrated with my collaborators, remember how I said they injected at 3 days? I had a very particular reason for being frustrated with them, and it has to do with a clinical trial that just got done. It was a big phase 3 trial by one of these really big stem cell companies that have a mesenchymal stem cell therapeutic specifically for stroke. So they're injecting the cells, not just the exosomes. We're injecting the bubbles. They injected the full-blown cells. Their data from their animal trials showed that their cells work the best up to 48 hours after the injection. After 48 hours, it starts to get a little wonky, right?
Okay.
What happens is when they went to design the clinical trial, the business people of that company said, you know what, we'll be able to reach way more patients if you just give us 1 more day. If you extend that time window, therapeutic time window, 1 more day to 72 hours. And guess what happened? The clinical trial failed because they chose the 72-hour. But when you went back and looked at the data, all the patients that received the stem cells up to the 48-hour mark looked good. And if they just would have made that their endpoint, it would have passed the clinical trial. But because all the patients at that last hour or that last day failed, it ended up killing the clinical trial. So when they injected my exosomes 3 days afterwards, the same that company did, I was like, forget about it. We're not going to see anything because they didn't see anything in the clinical trial. But yeah, we were still able to see a beneficial effect. So it kind of depends on the therapeutic. Just FYI too, the standard therapeutic for stroke, PA, it only has a handful of hour therapeutic window. So if you don't get the therapeutic in the first 4 hours, there's not a lot else they normally do for it.
One question I had is, so exosome are signaling molecules, signaling vesicles. So do you see, what sort of off-target effects do you see?
Say that one more time.
What sort of off-target effects do you see when just injecting an exosome?
Off-target effects? That is not uber clear. You're injecting something that's extremely complicated that has thousands of components. And I'm sure some of those things are desired, and some of those things, it's maybe doing a little bit of something that's maybe not desired. It's still sort of early days for the field, you know? So it will take a little bit more investigation. But on the whole, there's been a variety of these animal models. And in all the animal models, and in the one patient as well, it always seemed to have a net benefit effect. And if you do something called gross pathology, which is basically for, you don't do this with humans, obviously, but with your animal models, you basically cut the animal up into small pieces and send it to a pathologist, a physician that specializes in looking for signs that something has gone wrong with one of the tissues. So for instance, if you injected the exosomes systemically, one of the things you might see is your liver would help clear those out potentially. And so you'd want to see if there's any sort of pathology that's evident in the liver. And in all these, in the handful of studies that have looked at gross pathology, nothing's shown up. So it's probably context dependent on, you know, which disease we're talking about and where you get these bubbles from, but at least these exosomes from the MSCs seem to not do any harm. And so, incidentally, the mesenchymal stem cells that make the exosomes, they've been injected in like tens of thousands of animals and thousands of patients without any adverse effects seen. So it also makes sense that the bubbles they secrete, these exosomes, would also have a pretty strong safety profile. Yeah.
Okay, so when you said that you put the stem cells inside the heart for the cardiac muscle cells, all they did was cell-to-cell communication. If you put the exosomes in there, you would have gotten a different result, like greater result, more in this communication.
Well, the bubbles themselves are the communication signal. So one way to think about it, instead of me, this is probably a horrible analogy, but I'm going to say it anyway. So instead of me being up here and talking to you, and so when I talk, a little vibration in the air, you hear it in your ear. Instead of me injecting me around the heart and saying, fix yourself faster, it's like almost like they bottled me saying that, and they just, the bubbles themselves are the communication signal. So it's just sort of, instead of delivering the cell, which then creates a communication signal, they just injected the communication signals themselves. So one way to think about it is that it's a more concentrated form of what benefit it's having. Another little side benefit too is like when you inject cells into someone, a big fear, especially if it's a stem cell, is that although they've been injected in, like I said, tens of thousands of animals with no adverse effects, thousands of patients, no adverse effects, always the one fear that we have is it just takes one of those cells going a little rogue to get a little mutation in its DNA, and if the cell already likes to proliferate a little bit, maybe it could turn cancerous. Never seen that in any of the animal models or a human, but it's always a fear. So another little ancillary benefit would be if you can just inject these bubbles, the communication signals, you don't have to worry about that at all because it's not a cell, right? So, yeah.
So you mentioned that the exosomes can contain RNAs. I know that people sequence exosomes. Have you guys looked at how effective the RNAs that are present in exosomes are at actually translating into protein in cells?
No. Actually, the biggest area that people have looked at in terms of the RNA is microRNAs, which are super tiny RNAs that help control gene expression, right? And if you go through the literature, that's the vast majority of what people have talked about. And there's some reasons for that. There's been some high-up people in the field that sort of started that paradigm, but there's that excellent paper that came out last year, or a couple years ago, where someone quantitatively looked at, like, okay, well, you're saying these microRNAs, out of this complex mixture of thousands and thousands of things, you're saying this one microRNA is what mediated this whole effect? It's kind of like, So what this one paper, with this one lab did, and they published it in PNAS, which is a very good journal, they actually quantitatively looked at, well, how many microRNAs are in a given exosome, and it turns out that even not a lot. So in terms of the microRNA content, that one paper, because a lot of people have been hedging their bets on it being the microRNA content as the key drivers, and when that paper came out, it was right around the time that I was thinking about developing a collaboration, and that made me sort of rethink, hey, is it really the microRNAs we should be interested in? Because I really like non-coding RNAs. That's a little bit of my background, but that's what made me think to like, hey, maybe we should check out the proteomic, the protein profile of the exosomes, because not too many people are looking at that. They're just completely ignoring it. On the mRNA level, people can show that when you deliver the mRNAs, they're functional. It's very hard to piece out if you deliver something that's very complex with thousands and thousands of moving parts to pick out it was this one moving part that changed everything. It's likely a concert effect. Yeah, in a nutshell. Did I answer your question okay? Okay.
Well, we would, one more question.
Where and how do you source the exosomes? Like from the same species, same individual? What's the variation with that?
Yeah, so that's several questions wrapped into one. So we get the bone marrow from a patient, an adult. We get them from a company. They pay an adult because it's very painful. They stick a big old needle into their hip bone. They grind it around a little bit, pull out their bone marrow. And we have to get a good amount of bone marrow too, because these cells are very rare in your bone marrow. It's only like 1 out of 100,000 cells, somewhere around there. And then they profile that patient before they do that for if they have any sort of viruses, HIV, herpes, anything like that. And they also do an immune profile to make sure they're not sick or there's nothing wrong with them. And then once we get the bone marrow, we pull out these mesenchymal stem cells from adult humans. All of ours were from young male non-smokers on the East Coast, several ethnicities. So there's a lot of variation in there, ethnicity-wise. And then we grow them up. So go through a lot of cell division. So we have a lot more cells to work with. And then we just grow the cells more or less like normal, and then the cells will secrete the little bubbles into the media that we grow them up in. And then we just take off the media and then pull out the bubbles. So that media is normally just trash to most researchers. They just throw it away because it's really the cells that they're interested in. So we're taking what's ostensibly the trash, the media that has the bubbles in it, and just purifying out the exosomes and using them from there. And we do it in our GMP facility. So there's a good amount of, with all cellular therapeutics, there's a lot of variation from donor to donor. That's a very hot topic. Hey, how do we define what a good donor is versus, is there some sort of criteria with which we can measure the donor ahead of time? And the same will be true likely for the exosomes. There needs to be some sort of release criteria for them.
Okay, will you join me again in thanking Dr. Anderson for a wonderful presentation?
Thanks for sticking around.
It's interesting. I believe one of the hallmarks of a fabulous, wonderful research scholar is the ability to plan and have foresight. And I just want to thank you because in your presentation, you did your own biography. You also talked about the program, and that really made up for the fact that we lost 5 or 10 minutes at the beginning. So I personally want to thank you for that.
Yeah.
We have a student who would like to present you with just a small token of our gratitude for being here and something that you can hang on the wall.
Thank you.
Appreciate
Thank
it.
you for coming and speaking to us.
Looking
It was a
pretty
pleasure.
snazzy.
Thank you for being here.